Yeah, so up to this point, what have we done so far? Basically, we have figured out how to sequence an unknown pathogen. We figured out how to assemble that genome. We figured out how to annotate it. We figured out how to use a reference genome to speed up how we can collect genome sequences from new samples. We talked about multiple sequence alignments, so how can we actually line up all of these different genomes that we collect? And then last week, we talked about phylogenetic inference, which is basically, given the multiple sequence alignment, how can I use models of evolution to try to estimate the evolutionary history? So after we do what we did last week, now we have an evolutionary tree that shows kind of the relationships evolutionarily between all of these different viral genomes that we've collected. But the problem is that the branch lengths along the genome that we collect, along the phylogeny that we have inferred, are a unit of like mutations. It's basically how many differences in the sequences are there between any given two sequences. So kind of those branch lengths on that evolutionary tree, they're a unit of mutations rather than time. So today, what I'm going to talk about is what are some tasks that we can do using that evolutionary tree that we've constructed. So we're going to first talk about ancestral state reconstruction, which is basically a way of kind of inferring ancestral sequences. We're going to talk about dating of phylogeny. So I mentioned the branch lengths are a unit of number of mutations, but we ideally want them to be in unit of time. So we want those branch lengths to be like, oh, the common ancestor between these two viral samples was a week ago or a day ago, right? Not 2 mutations ago or whatever that would be. So we're going to talk about how we can scale a phylogeny, how can we scale an evolutionary tree to now be in unit of time. And then we're going to talk about how do we assign genomes to lineages. So you might have heard of like Delta variant, Omicron variant, all those kind of lineages that have been coming up for COVID-19. We're going to talk about, if you gave me a genome sequence of a COVID-19 sample, how do I assign it to a lineage? And lastly, we're going to talk about some of the kind of intro epidemiology related stuff of how do we study the prevalence of mutation. So first, ancestral state reconstruction. Remember that, imagine this is the true evolutionary history. So I had some ancestor and then that mutated as we go down the tree. And then kind of each mutation is inherited all the way down. And remember that in reality, we can't actually see the evolutionary tree. But as of last week, we've come up with a way of inferring the maximum likelihood or the most likely evolutionary tree. So as of last week, we can come up with this structure. And now our question is, how can I actually reconstruct the mutations that occurred along the specific branches? So far, all I have is the structure of the tree, and I have the branch lengths, but I don't have the actual mutations that occurred. So I don't have the sequences of any of these ancestors. So how can I reconstruct those ancestral sequences? How can I reconstruct which specific mutations occurred along each branch of the tree? And this is a computational problem known as ancestral state reconstruction. So much like how we did maximum likelihood phylogenetic inference, so we defined a probability function and we maximized that function over all the spaces of trees. That was what we did last week. We can take a similar approach to inferring where those mutations occur along those branches. So yeah, this is called maximum likelihood ancestral state reconstruction. And it's actually an algorithm that's very much like that Felsenstein's tree pruning algorithm I mentioned last week, that kind of bottom-up approach for computing the probability of observing a given collection of sequences given the tree structure. So what we do is what's called a post-order traversal of the tree. If you ever take like CSE 12 in the future, you're going to learn about tree traversal algorithms. But basically, a post-order traversal just means we always will look at any given node after I've already looked at its children. So basically, I'm going to visit all of the nodes of this evolutionary tree, but I'm going to visit the leaves first. And then as any given node will only be visited after I've already visited its children. So if the current node is a leaf, I already know its sequence, right? In my multiple sequence alignment, I know the sequences of all the leaves. So in my kind of bottom-up traversal, if the current node that I'm looking at is a leaf, the sequence is defined. We were given the sequence. Otherwise, for any of the internal nodes, what I could do is use that model of evolution that I inferred. Like, remember from last week, we talked about all those models of evolution. I can use that model of evolution for each position of the multiple sequence alignment. I can see what is the most likely symbol at the current node, given the symbols that the children have at that position. So for example, if at the first position, both my children have a C, I'm going to say my symbol is also a C. Versus if both my children have an A, I'll say, my symbol's an A. If one of them has an A, one of them has a C, I'm not sure, but maybe I'll just kind of guess 50-50. So we can kind of model the uncertainty of each of those letters as well. And the result is what we can essentially do is kind of label our tree with the best guess at each position. So for example, in this tree here, right, imagine I didn't see these internal sequences. What I could see is, well, at the first position, I have an A and a C. Okay, I'm not too sure which one's the true sequence. Here I have a C and a C. Well, that one's definitely a C. Here I have a C and a C. So that one must have been a C. And here I have a T and a T. So that one should be a T. And the only one I was uncertain of was this A over here. But kind of as I reconstruct information from the rest of the tree, I can see that, well, everything else has an A in that position. So maybe an A makes the most sense to put here as well. And kind of keep going, I can see that like in this internal node, I have an A and an A. So I'm going to guess that that's probably an A. I have a C and a G. So I'm not sure what the ancestor was here, but kind of looking elsewhere in the tree, I can see, well, yeah, it seems like most of my sequences have a C in that position. So it maybe makes the most sense for this to be a C. This gap, both of them have a gap there. So I'm going to put a gap, and then T and T, I'm going to put a gap. And then when I'm reconstructing this one, I see the first position is A and A. Okay, so that's probably an A. C and C. Okay, so that's probably a C. C and gap. Well, this one, I don't really know what my ancestral sequence was. I actually wouldn't have been able to reconstruct this G. So this one is actually, I would have probably either guessed a C or a gap. So this one, I guess maybe I should have changed in the example. And then T and T, I would guess this is a T. But basically, I can attempt to reconstruct the sequences at the internal nodes. Cool. Okay, any questions about that? Basically, using the sequences at the leaves, I can try to kind of predict upward. Questions kind of make sense? All good? Yeah, cool. So this is especially important if, for example, I want to know, if there's some variant that just came out, I want to be able to say, hey, this mutation that's really important for how this variant infects our cells, where exactly in the evolutionary tree did that mutation come about? So now we can answer those types of questions. Okay, so the second topic we want to cover today is called dating of phylogeny. So as I mentioned, when we infer a phylogeny, so when we give a multiple sequence alignment to one of those tools that we talked about last week, the tree that we predict, the branch lengths are in unit of number of mutations. So I can basically answer the question, how many mutations occurred from the parent to the child in that evolutionary tree? But as a human, right, I want to deal with time. I want to be able to answer questions like, when exactly was the ancestor of these two sequences, or how long ago was the common ancestor of all COVID-19 sequences, right? So if these branch lengths were in unit of time rather than mutations, I could answer questions like, how much time has passed from parent to child? So using the techniques that we talked about last week, I can infer a phylogeny where the branch lengths are in unit of mutations just using the sequences. But if you also gave me information about time, I can use that information to try to scale those branches such that I come up with branch lengths in unit of time that line up with that time information. And the most common time information that we're going to use is sample collection dates. So for example, on campus, let's say that you get one of those nasal swabs from the vending machines, and then you do the nasal swab, you submit the collection into the bin, and then they collect it at the end of the day. Every single sample they know was collected on that specific day. So if they then later sequence it and then come up with a genome sequence, then infer an evolutionary tree, the leaf that corresponded to you, we know what the collection date was. We know exactly what day that sequence was obtained. So using that time information, we can try to scale those branches. And what we're effectively doing by doing that is we're effectively estimating mutation rates along each of the branches. So we're basically trying to estimate the number of mutations per time, right? A rate is something per time. Mutation rate is mutations per time. And if we can estimate mutations per time for each of those branches, we can kind of scale the branches accordingly. Does that idea kind of make sense? Ish. Cool. Okay, nice. So there's a lot of tools that are used to do this. A common one is called tree time. And here's a GitHub repo. I think it's implemented in Python, so it's fairly kind of simple to open up and take a look at. A bonus of this tool is that it can also do ancestral state reconstruction. So if you gave this tool your evolutionary tree that had mutation, like the branch lengths were in unit of mutation, and you also gave it the time information, it'll automatically do both. It'll do ancestral state reconstruction and scale the tree to be a unit of time. There's another tool called LSD2, and here's the GitHub link. So this tool uses a technique called least squares dating, where it's basically trying to minimize like the squared distance between like the estimated time of any given leaf and the true time that we've known to collect. So it's taking this like least squares dating approach. And then there's a less popular tool, but it's fast and therefore somewhat interesting, is this tool called logdate, that it basically is trying to minimize some deviation of the sample times after you scale it. So yeah, so all of these tools, they're all kind of solving the exact same problem, which is just how do I scale this tree to be a unit of time? But each of them is making different model assumptions and kind of taking a different approach to that problem. And as a result, they have kind of trade-offs between accuracy, runtime, kind of all that good stuff. Cool. Okay, so the next thing that we can do with our genome sequences, now that we have this evolutionary tree, is assign genomes to lineages. So what is a lineage? This word gets thrown out quite a bit. A lineage is just a subtree of the overall phylogeny. So I have my overall evolutionary tree of all the samples I've ever collected. A lineage is just a subtree of that evolutionary tree. So biologically, we can think of a lineage, right? So remember that mutations are inherited. We can think of a lineage because it's a subtree. We can think of it as all viral sequences that inherited all the mutations from the root up to that point. So from the common ancestor up to kind of the top of that subtree. Then there's also this term strain that gets thrown around quite a bit. And a strain is a significant deviation from the ancestral virus. And I actually tried looking into what do they mean by significant deviation. So that definition was from a CDC website, but I couldn't actually find a very firm or like specific definition of like what constitutes significant deviation to make a new strain. But in general, the overall sentiment that I'm seeing is that it typically seems to be some notion of like there was a significant functional or phenotypic difference. So basically, somehow a bunch of really smart virologists have determined, okay, this virus has now changed so much that this phenotype or this function is now significantly different. But it seems to be more of kind of like a categorization thing rather than like a strict set of rules or anything like that. Whereas a lineage is a very specific, you know, you are this subtree of the evolutionary tree. So for SARS-CoV-2, so for the virus behind COVID-19, people use this tool called Pangolin to assign genomes to lineages. And this tool is open source. It's actually a really interesting tool, and they have a nice web app as well that you can just kind of submit a sequence to. But basically, what Pangolin does is in advance, a lot of virologists have kind of accumulated and noted what are a bunch of common mutations that we see in different lineages of the virus. So they have like this global evolutionary tree, and they kind of see what are some common mutations that different lineages have. And then Pangolin takes that information and it takes an approach known as a decision tree. A decision tree is like a very simple machine learning approach. And given a sequence that you provide it, it will use a decision tree that was trained on the global data set to try to assign your sequence to a lineage based on which mutations your sequence has. So it works really fast, and it's really useful for kind of like categorizing new sequences that you see. Cool. Any questions so far? Kind of makes sense. Yeah, and then once we have Pangolin, now we can kind of do things like, if I have a bunch of COVID sequences from UCSD, I can assign them to like Delta or Omicron or whatever lineage they came from. And then we can see how are the different lineages kind of growing or falling over time. And that is the focus of the next part. So studying the prevalence of mutations, this is kind of, now we're entering the really hands-on public health related aspects of what we've been doing. So mutations are important because, you know, if one mutation happens to be growing significantly in a population, that could potentially imply that it somehow is making the virus more infectious. So knowing which mutations are becoming more prevalent in the population can help you with like vaccine design, with therapeutic design, you know, all that kind of stuff. So one common tool for studying the prevalence of mutations is NextStrain. And basically NextStrain allows you to visualize the evolutionary tree plus the mutations plus demographic data. And I think I might have shown NextStrain a few weeks ago, but kind of it creates visualizations like this that maybe at the end of class I can also tinker around with to show you. But basically what it shows you is a map that shows all of the different lineages that are showing up in all the different countries around the world. And then it also has this evolutionary tree of a bunch of the genomes that they've collected. And it also measures how many mutations are occurring at each position of the genomes. So this plot over here, the horizontal axis is position of the genome. So this is the beginning of the genome. This is the end of the genome, right? SARS-CoV-2 has like 29,000 letters. And then on this axis, the vertical, on this plot, the vertical axis means diversity. It means deviation, or kind of like how many, you know, how much deviation is there at this position? Like how many different letters do we observe at each position? So a higher diversity means we have more mutations, more different letters at this position. So with this, what we can kind of see is what parts of the genome are very kind of conserved, and conservation usually has something to do with fitness, versus what parts are not very highly conserved. And this can very directly inform vaccine design, because what you would want to do is design a vaccine that targets a region of the genome that is relatively stable, right? Like waning vaccine efficacy, one of the main mechanisms that vaccine efficacy drops is you designed a vaccine for a specific part of the virus. Your body builds antibodies that can target that specific part of the virus, but then the virus mutates, and now your antibodies can no longer recognize the newly mutated versions of the virus. So if we can have a vaccine that targets a part of the virus that doesn't change very frequently, hopefully our antibodies that we develop will still be able to recognize that portion of the virus. So yeah, we can play around with NextStrain a little bit later. There's another tool called covariance that tracks SARS-CoV-2 variants and mutations over time. And you can find it at covariance.org. This is actually run by a research lab over at Scripps Research Institute, which is kind of really local. And what's really cool is that you can actually overlay, you can kind of see these plots for a bunch of different countries around the world. I think they also have like county level data where you can see what is the prevalence of each of the variants over time. So you can kind of make inferences like, hey, this orange lineage, so whatever this 20E lineage was relatively small, but very quickly dominated. But then once this red lineage showed up, this like 20H, I think maybe that color matches up, maybe it's this one, I don't really know. It seems like that one quickly dominated. And then at the end, we can see that, hey, maybe this green one is going to dominate next. So using this information, this is how we can detect things like, hey, Omicron has now, become significantly dominant in San Diego County. We can make plots like this to kind of see what is the prevalence of each of the variants over time. And the last one that I want to point out is outbreak.info, which is also, I think Outbreak is also run by a lab at Scripps Research. Actually, I think Outbreak is the one by Scripps Research. I think covariance is from a different group. But outbreak.info, what's really cool about it is that it has a bunch of public data that show you not only like mutation information, but it also shows you things like prevalence across the world. It has a bunch of statistics on cases, deaths, how many tests are being performed, all that kind of stuff. And it has these really useful like paragraphs kind of in a human readable way interpreting the data. So the other ones, they're a lot more useful just to kind of see the raw data, but this actually is very useful for kind of communicating the important information. And what's really nice about this database, outbreak.info, is that they have an API, so you can actually write programs that can pull data from outbreak.info very easily. So if you want to make your own visualizations or you want to do your own large-scale research analysis using their data, it's very easy to kind of pull their data and tinker with it on your own.